Hello students, a warm welcome to all of you in BioDesire. In today's session, we discuss about the biochemistry. In today's session, we discuss about some basic concepts of biochemistry. So we start today's session and understand first topic, that is the biomolecules. What is the biomolecules? Okay, what is biomolecule? Biomolecule is an organic compound that are produced by the living organisms, and these biomolecules are very essential for the structure of your body, for performing the different tasks in your Now we understand what is the biomolecules. What is the biomolecules? Biomolecules is the organic compound that are formed in living organisms. Okay, living organisms, and these are the different So first we discuss what is the biomolecule. What is the biomolecule? Biomolecules is the organic compound or organic molecules that produced by the living organisms, and these biomolecules are very essential for structure of for the structure, for the function, for the growth, and for the metabolic pathway, and regulate the life process of living organisms. Without these biomolecules, we can't image our life. Okay, because each and every metabolism pathway, this biomolecules plays a most important role. Okay, and this biomolecules is made up of carbon, hydrogen, oxygen, nitrogen, phosphate, and sulfur. And this molecules, whenever these compounds are mhm, mainly this biomolecules is made up of this carbon, hydrogen, oxygen, and nitrogen, phosphorus, or sulfur. Okay, and the example of biomolecules is DNA, RNA, protein, carbohydrate, lipids, fats, protein, etc. Vitamins, etc. Okay, what is the biomolecule? Biomolecule is equal to the molecules of life. Okay, so first we discuss about the carbohydrate. This carbohydrate is a very essential and very important biomolecules. This biocarbohydrate is a source of energy. Is a source of energy. And without energy, a living organism can't survive. Okay, that's why this carbohydrate plays a That's why this carbohydrate plays a most important role. And what is the general formula of carbohydrate? General formula of carbohydrate is a CH2ON. Okay, N is your number of carbon. Means let's suppose your sugar is a 3 carbon, then C3H6O6. If your carbon is N number is 6, then C6H12O6. And this is the molecular formula of glucose, and this glucose is very essential for the generation of ATP. Okay, and this carbohydrate is divided into two parts. First is the base of aldose group or keto group. Aldo group means CHO, keto group means this one. Okay, polyhydroxy aldehyde or ketone. Okay, now we discuss about the functions and example of glucose. Now we understand the Now we understand the function of carbohydrate. Energy of source, glucose. Energy of storage, glycone or starch in plants. Structural role like chitin or cellulose. Cellulose in plant, chitin in fungus. Cell recognition like glycoprotein or glycolipid. Nucleic acid component like ribose sugar or derivose sugars. Okay, these are the sources and these are the example of their function. Okay, now we discuss the classification of carbohydrate. Basically, this carbohydrate is divided into different types. First is your monosaccharide, whenever the single sugar unit like glucose or fructose. Disaccharide, whenever two monosaccharides are composed to each other, then it will form disaccharide. Okay, like example sucrose or lactose. Oligonucleotide, when 3 or 10 monomeric units are connected to each other by the help of glycosidic bond, then it will form the oligonucleotide. An example is of raffinose. Polysaccharide means whenever the n number of monomeric units are connected to each other, then it will form the polysaccharide. And the example is starch or glucose. Glucose is made up of cellulose is made up of n glucose. Whenever the n number of glucose are connected to each other, then it will form cellulose, and same for the starch. Okay, monosaccharide means n number of chain. Disaccharide means only 1 unit of sugar. Here 2 units of sugar. Here approximately 3 to 10 unit of sugar, and here the n number of sugar. Okay, monosaccharide is a very simplest sugar. Okay, this monosaccharide cannot be hydrolyzed further, but this disaccharide, oligosaccharide, polysaccharide can easily hydrolyzed. Okay, now we discuss about the classification of sugar on a basis of functional group. On the basis of functional group, this carbohydrate is divided into two parts. First is the aldose sugar, second is the ketose sugar. Okay, and the example is CHO, that is the aldehyde group, and C double bond O, it is the keto group. And the example is glucose, ribose, and fructose. Fructose is a type of keto sugar. Glucose is a type of aldehyde sugar. Okay. Now we discuss the classification on the basis of carbon number. Whenever n equal to 3, now it is known as a triose. An example is a glyceraldehyde. Whenever the n equal to 4, then tetrose. An example is erythrose. N number equal to 5, then pentose. An example is ribose or deoxyribose. N number equal to 6 means hexones like glucose or fructose. N equal to 7 means heptose or septoheptose. Pseudoheptulose. Okay, for general formula for this is CnH6O3. For tetrose, general formula is C4H8O4. For pentose, C5H10O5. For hexose, C6H12O6. For heptose, C7H14O7. Okay, these are the normal formula. This is the general formulas. Okay. Now we discuss about the isomeric nature in monosaccharide. Okay. This monomers or this monosaccharide shows the different types of structural and stereochemical isomerase due to the chiral carbon. Okay. Means in this carbon, in 4 different groups are present. That's why it will show the stereochemical isomerization process. Stereomerization means same molecular formula, same structural, but different in spatial arrangement. Only arrangement is different. And the example is L or DL isoform. Okay, LD and L isoform. These are the two types of isoforms. First, right hand, D means right hand, L means left hand. Okay, and the example is glucose, L glucose, D glucose or L furose, fucose. Okay, these are the examples. Formula is same, but conformational is changed. And on the basis of this is divided into two types, D form or L form. Okay, D form is right side and L form is left side. Right side, this side, this rotation is this side, then D, rotation is this side, then it is known as L glucose. These are the two conformation of, this is the two isomeric form. Structure is same, only conformation is changed. Okay, now we discuss about the epimers. What is the epimers? Whenever the two monosaccharides are different configuration at only one specific position. Okay, only one position is changed. Like in one position H and OH. At the fourth number of carbon, this is present like this. Now this conformation is changed and this condition is undergo the epimers, and the example is glucose and galactose, glucose or mannose. Correction is changing at the carbon number 4 or carbon number 2. Okay, this is the epimers. Now anomers means isomer formed after the cyclic structure formation due to the conformational changes in anomeric carbon. Okay, these are the alpha or beta. Alpha means OH is present an up downside like this side, and beta means OH group is present at upside. Okay, this is also known as a transform or trans or cis form. Okay, like alpha D glucose or beta D glucose is the example of anomers. Now we discuss about the mutarotations. Mutarotation means interconvention between the alpha or beta anomers in a solution by an open chiral carbon, and changes in specific optical rotation until equilibrium is reached. Example is alpha D glucose rotation or beta D glucose rotation at this angle. Okay, and maximum rotation at this angle. Okay, mutarotation occurs only in molecules that have a free anomeric carbon. Okay, reducing sugar can be mutarotated. Okay, these are the mutarotation. Epimers, anomers or isomers. Isomers means only direction is changed but structure is formed same. Epimers means only one carbon is changed, varied, then it is epimer. Anomer means whenever we convert into cyclic form, then due to the anomeric form, it will change some configuration like alpha or beta. Mutarotations means interconventional between a alpha or beta anomeric chain. Okay. Now this is the quick revise table. D and L means orientation. Example, glucose. This is the most natural sugar in mostly natural sugars are present in D form. Epimers means only one chiral carbon is changed. Example is glucose and mannose at C2 carbon. Anomers means differ at anomeric state like alpha versus beta glucose formed during the ring closure. Beta rotation means alpha shift to the beta, beta shift to the alpha. It is a shifting concept. Optical rotation change in glucose or in reducing sugar. Okay. Now we discuss about the disaccharide. Disaccharide means whenever two monomeric units are linked to each other, then it will form disaccharide, and this is the glycosidic bond. And example is sucrose, lactose, malactose. And mannose, glucose, fructose, glucose, galactose, glucose, glucose. This is the combination. Whenever the two molecule of glucose, whenever a glucose and fructose are combined to each other, then it will form sugar. Whenever the glucose is combined with galactose, then it will form lactose. Whenever the two glucose are connected to each other, then it will form maltose. Okay, and the bond is this one. These are the glycosidic bond. And this one, first one is not reducing sugar, and second and third is a reducing sugar. Okay, now we understand the concept of reducing and non-reducing sugar. What is reducing sugar and what is the non-reducing sugar? Now we discuss about the reducing and non-reducing sugar. What is the reducing sugar? Reducing sugar is a carbohydrate that has a free aldehyde group or keto group, either in linear or in open chain, and can easily reduce via the oxidizing agent. Okay, reducing sugar contain a free functional group. Free functional group like aldehyde or keto. Okay, they can be mutarotation. Okay, and this also hydrolyze easily reduced by the help of oxidizing agent, and it will also give the positive result in Benedict result, in flying result, or in Tolan test. And this reducing sugar also give the positive result in Benedict test, Fehling test, and Tolan test. Okay, why are they reduced? Because they are the free anomeric carbon. Okay, and this anomeric carbon can open into the linear form, and aldehyde and keto is easily oxidized. Okay, now non-reducing, and the example of reducing sugar like monosaccharide like glucose, fructose, galactose, mannose, disaccharide like lactose, polysaccharide like glucosan or starch. Fructose is ketones, but still reducing. Okay, fructose is a keto, but it also is reducing agent. It will convert to glucose or mannose under the alkaline condition. Okay, next we discuss about the non-reducing sugar, and this non-reducing sugar does not contain a free anomeric carbon. That's why it is not reduced by the help of oxidizing agent. Okay, because why this will not contain the anomeric carbon? Because carbonyl group is involved in glycosidic bond formation. Okay, means this functional groups also participate in a formation of glycosidic bond. That's why this sugar is a non-reducing sugar. Okay, and cannot open in linear form. Do not reduce. Do not re, and do not reduce by the reducing agent like Benedict. For Tolan, it will give the negative result and cannot go under the muter rotations. Okay. Now the example is sucrose, trehalose, and raffinose. Sucrose is also known as the most commonly reducing sugar. Okay. Now we discuss about the key differences in reducing sugar or non-reducing sugar. Reducing sugar contain the free anomeric carbon, and non-reducing sugar no. Muter rotation yes, no. Benedict and Fallon test positive, here negative. Open eating chain yes, and here negative. An example is glucose and galactose, here the sucrose and trehalose. Okay, this is the reducing and non-reducing sugar. Okay, now we discuss about the oligonucleotide sequence. Oligosaccharide. Oligosaccharide means whenever the 3 or 10 monosaccharide are combined to each other, then it will form the oligosaccharide. And this is oligosaccharide is present in glycoprotein or glycolipid. An example is raffinose or mannose. Next is menan in yeast or plants. Okay, role to cell recognition or immune response. Okay, now we discuss about the polysaccharide. Okay, polysaccharide means whenever the n number of units are combined to each other, n number of monomeric unit are combined to each other, then it will form the polysaccharide. Okay, now we discuss about the classification. First classification on the basis of storage. On the basis of storage, this is also divided into different types like and the example of storage polysaccharide is starch and glucagon. Starch in case of plant and glucagon in case of animals. Starch means amylose and amylopectin are linked to each other and it will form the plant storage. And glucagon, it is a highly branched structure whenever and it is a stored material in plants. Okay, glucagon more branched than starch. Okay, now on the basis of structure, like it will homosaccharide, homopolysaccharide, homopolysaccharide means whenever the same n number of monomeric units are combined to each other. Okay, like cellulose. Cellulose present in plant cell walls. Here the n number of glucose are combined to each other. Next is your chitin. Chitin means here n number of and acetyl glucosamine are combined to each other and it will form chitin. And then the example is fungus and arthropoda. Okay, and the bacteria and the humans cannot digest the cellulose. Okay, because it will lack of cellulase enzyme. That's why this animals can't degrade the cellulose. Next is the heteropolysaccharide. Whenever the mixed number of or n number of monosaccharide unit are combined to each other, then it will form the heteropolysaccharide. An example is hyaluronic acid like glucuronic acid or aceto. Example is hyaluronic acid polypeptide on heparin. Okay, and this is the composition and this is the example. Okay, this is the heteropolysaccharide. Okay, this is heteropolysaccharide, structure polysaccharide, oligosaccharide, reducing sugar, non-reducing sugar, monomers, epimers, and this is all about the carbohydrate. Okay. Now we discuss about the protein. What is the protein? Protein is the biological macromolecule and it is made up of amino acids. Whenever the two amino acids are linked to each other, whenever the n number of amino acids are linked to each other, then it will form the protein. And one amino acid are connected to other an amino acid by the help of peptide bond. Okay, and this protein is a structural and functional backbone of our cell. Okay, and this polysaccharide is made up of carbon, hydrogen, oxygen, sulfur and nitrogen. Okay, proteins one and more polypeptide chains are combined to each other, then it will form whenever one or more polypeptide chain are fold into a functional 3D structure of your protein. Okay, now what is the building block of protein? The building block of protein is a amino acid. There are the 20 amino acids are present. Some are essential, some are inessential. Okay, and two polypeptide chains are combined to each other by the help of peptide bond. Okay, unlike the condensation means loss of water. Okay, next is the Zwitter concept. Zwitter concept means amino acid exists as a dipolar ion at a biological pH. Now we discuss about the Zwitterion concept. Now we discuss about the Zwitterion concept. In Zwitterion concept, whenever the net charge on your protein is zero, then that condition is known as the isoelectric point. Now we discuss about the Zwitterion. Zwitterion means whenever your compound or protein contain the dipolar ions like plus ion and minus ion, both the number is equal, then it is known as a Zwitterion effect. Okay. Next we discuss about the now we discuss about the labels of protein. The label the protein is divided into four types. First is your primary, secondary, tertiary, and quaternary. What is the primary structure? Primary structure is a linear chain of amino acids, and one amino acids are linked to each other by the help of polypeptide chain. Okay, and this is very helpful to determine the identity and functional. Okay. And for example is insulin structure, and this structure is discovered by the Fritz Sanger, and they also got the Nobel Prize on this novel research, on their novel research. Now structural, on the secondary structure of protein, whenever the primary structure is compact and form the alpha helix and beta sheets, then it is no beta sheets, alpha helix, beta sheet turns and loop, then it will form the secondary structure of protein. Alpha and okay, and these complete structure is established by the hydrogen bond. Primary structure is stabilized by the disulfide bond, and secondary structure is stabilized by the hydrogen bond. Okay, alpha helix means ring-handed figure. Okay, and like example is hair protein or keratin. Beta plated seated means chain side by side like silk. Beta turn means reverse direction folding, means proteins will tightly turn. Okay, now we discuss the hmm. Alanine is known as the alpha maker and taurine is known as a alpha breaker. Okay. Now tertiary structure. Tertiary structure, whenever the secondary structure is compact by the help of hydrogen bond, hydrophobic intergen, ionic bond, van der Waals and disulfide bond, then it will form the tertiary structure of protein. And this tertiary structure of protein is a fully functional unit. Okay, and the example is myoglobin. Okay, now quaternary structure, whenever two polypeptide chains are combined to each other, then it will form the quaternary structure of protein. Okay, and the example is hemoglobin. In hemoglobin, four subunits are present. Okay, now we discuss about the classification of protein and their composition, like simple protein, like only amino acids. Example is albumin. Conjugated protein, whenever the protein are combined with non-protein compound. Like derived protein means form the hydrolysis, like peptides. Okay, shape like fibrillous or global. Fibrous means in long or starch, or globular means compact and functional. Example is keratin protein, collagen protein, enzymes and halogens. Okay, these are the classification of protein on the basis of shape. Now we discuss about the function, like enzymatic. For enzymatic activity, protease for structure, collagen for transport, hemoglobin for hormonal, insulin for immuno, antibodies for contrastal, actin in the myosin, and storage by the help of ferritin. Okay, these are the different types of proteins or their, and this is the function or example. Okay.